It's my extreme pleasure to be here and to look out into the audience and see such successful and well-qualified scientists, both early and late career. And so it's my honor to present this information. I'm going to try to cover the progress we've made using GWAS and poplar genomics in identifying key genes associated with important economic traits, and then how we're trying to move those into the market and how we hope that they'll impact the future of biology. We're standing here at the beginning of an incredible next 50 years. I wish I would be around for the next 50 years. Biology is going to change the way we live, the way we manufacture, the way we heal ourselves. And you all are really going to drive that forward. And so the next 50 years is going to be known as the age of biology. We humans do the easy things first. It's just our nature. And so the first easy thing we did was physics. And the second was chemistry. The more challenging science is biology. Biology is more complicated than physics, more complicated than chemistry. I have a chemist sitting in the audience here who's shaking his head. John, chemistry is great and very, very important. But biology is more complicated because it infuses all of those things. And we use genomics to try to tease apart what is controlling the way we grow, the way a tree performs under stress. And I hope to show you some examples of that. I'm going to cover the sequencing assembly of the poplar genome, creating the genome-wide association study that Steve so nicely introduced, some of the discoveries, and a look to the future. So, poplar's earliest progenitor occurred 470 million years ago, when the first aquatic plant moved out of the water and onto the terrestrial ecosystem. This was a very simple plant. It contained all the future opportunities that we see now in poplar or Norway spruce or Scots pine. It didn't have all the anatomies or features that we see in trees today. It did have photosynthesis, that evolved earlier, but the environment in which this first progenitor evolved was very different than the environment we live in today. And in fact, that event, that first terrestrial plant, which led to all land plants, may have happened right here on the spot we're standing. 470 million years ago, Sweden was in a subcontinent called Baltica. And Baltica was at the equator. And that was the proper environment for where the evolution of land plants may have occurred. Now, this first land plant didn't have roots, didn't have complex cell walls, it didn't have a vascular system, but it succeeded. And it had a toehold on land, which ultimately led to trees. Poplar entered the human record with the Romans. The Romans recognized that you could take a dormant stick of poplar and then drive it in the ground, and it would root itself, and it would then grow from that unrooted stick. And they distributed these dormant cuttings to the populace, to the people. And that's where the genus comes from. It's the people's tree. That's what it's known as. And so the Romans used it to line their boulevards and to create urban environments that were attractive to people. And so Napoleon then adopted that same strategy, although his intent was to march his armies into Prussia and conquer Europe. And in the winter, his armies got bogged down in the snow because they couldn't see the road. And so he used this technique of dormant cuttings to line the streets so his armies could move in the winter. This environment led to many sketches. That's a Van Gogh sketch. And so we can see those poplar-lined boulevards even today. To be more centric to the U.S., Lewis and Clark, who explored the western U.S., used cottonwood to create canoes that allowed them to navigate the rivers. And then, of course, famous battle that led to the United States having Texas as part of its statehood. Alamo in Spanish means poplar. And there was a large cottonwood tree planted in the middle of the mission that then led to the defeat of the Texans by the Mexican army. We began looking very early at using poplar to inform biology. And Tongming Yin here was a postdoc in my lab. He's now the vice president of research for Nanjing Forestry University. And we used microsatellite markers, SSRs, that Steve highlighted, to identify chromosome 19 as the chromosome that contained the genes that determined gender. Poplar's dioecious, meaning there's male trees and there are female trees. And in a breeding program, what we found was that the vast majority of the superior genotypes were male. And if every one of your selected genotypes were male, there was no future generation. So we wanted to identify early markers, so when you're selecting a tree that has not become reproductively mature yet, you could get a balance of males and females. So that was the practical application of this. And we were able to do that very early on without the genome. We could identify markers that would then allow breeders to say, I want 50% females and 50% males in my breeding population. Well, just about that time, the ability to sequence genomes came about. And collaboratively, we worked with many, many, many people around the world, 100 co-authors on that paper. And the folks in Umio Plant Sciences were key to helping us bring that together. Stefan, Ulve, and Rishi all contributed to gene annotation. At that time, Umio had the world's best expression or RNA library, RNA being the messenger of information in the genome. And so they were willing to take that unique resource and fold it together with the sequencing to allow us to annotate or to identify genes in the genome. So very critical. At JGI, I worked with Jeremy and Dan, and we began pulling together the poplar genome. This was the first genome, plant genome, to ever be sequenced, assembled, and annotated using shotgun sequencing. Everything before that, humans and Arabidopsis, were done by bacterial artificial chromosomes, back tiling. So you would get a piece of DNA and you just build this tiling alignment. And it took years and years and years to do that. But Craig Ventnor at the Ventnor Institution said, why don't we just chop up the whole genome and then sequence the little pieces, and then use computational sciences to rebuild the chromosomes. This had never been done in plants. So we proposed it, the US Department of Energy agreed to do it, and so we were the first to ever use shotgun sequencing. That's the technique that's now been used in every other genome, up until today where you have long read sequencing. We used genetic maps that you saw on the previous slide to build these chromosome assemblies. And from that, we were able to depict the evolutionary history of how did the genome come into being. And it turns out there was a whole genome duplication called the salicoid duplication depicted in that complicated cartoon you see behind there. It was we had, let me say, when we did the shotgun sequencing, we had about 150 pieces of the genome. And we didn't know how they all related to each other. So I spent weeks and weeks trying to put that together computationally and got very frustrated. Sat literally for just a few minutes, rendered my thoughts, asked for help, and all of a sudden that entire genome revealed itself to me. So inspiration or divine intervention, I don't know what it was, but there was this moment where the genome became clear, and we published that. It was also, we knew here, comparing the Arabidopsis duplication with the salicoid duplication, that the molecular clock in poplar is ticking slower than it is in humans and than it is in Arabidopsis. So we think of DNA mutations accumulating at a constant rate, and you can use that accumulation of mutations to set an evolutionary timeline. How long ago did two species diverge? And it turns out that poplar's clock is ticking at a different rate. And the reason that is that you have this long-lived perennial plant that has the ability, when a branch breaks off and it floats down the river and embeds in sand, to germinate. and grow and mature. So individual genotypes, not individual trees, individual genotypes can live for thousands of years, 10,000 years, 15,000 years, and those ancient genotypes are casting those ancient gametes into the gene pool constantly. And so every time that event was successful, the molecular clock shortened. And so poplar looks like it's about 1/3 the age that it is evolutionarily. And that's important because it tells us that we have the opportunity to take advantage of that molecular diversity in our breeding programs. So it accumulates this diversity outside of natural selection. And then that diversity is available to us to modify lignin or to find genes for disease resistance. And that's what we did. We began to look for variation in individual markers associated with DNA. Well, Jeremy and Dan and I continued to sequence genomes. We sequenced foxtail millet, brachypodium, kalanchoe, and then ultimately eucalyptus, and you'll hear more about that from Dario later on. Steve DeFazio entered the scene at that point. We had this rich genomic resource, but we wanted to create a genome-wide association population. And Steve mentioned that this is done in humans quite a bit to link traits to genes. So to do this, we collected 1,500 genotypes along the western coast of North America and resequenced all of those. So now we don't just have a genotype, we have 1,500 genotypes. And then because of this propagation system where you can take a dormant stick, pop it in the ground, we clonally replicated that population 4 times in alternate environments. So now you can begin teasing apart, does the gene express itself only in wet environments, or is the gene expressed in all environments? And that's the power of common garden and association studies. We developed a SNP library. Steve and I began working with isozymes. You'd have 24 markers, you might have 50 markers. We moved to rapids where we had hundreds of markers, but with SNPs, single nucleotide polymorphisms, we had 65 million markers. So now we've saturated the genome. Every 17 base pair across 480 million bases has a variable marker. And this is unprecedented in biology. It's better than the human mapping population. It's better than the corn or soybean mapping population because now we can look at individual SNPs, those DNA variants, and link them to protein structure. A single change in a nucleotide, ATGC, can lead to an amino acid substitution that then can lead to a phenotype that's valuable to foresters deploying this ultimately in the field. From this, we associated every phenotype we could measure and found 35 genes that went into invention disclosures. There were 16 patents issued out of a collective work over the last 20 years, and 6 of those are in licensing agreements, and I'll tell you a little bit about those. So I had the pleasure of working with Tim Czaplinski, who is a plant biochemist, physiologist, and he has developed the world's leading library of metabolites. Metabolites are small compounds, secondary metabolites, that have impacts on plant performance. We heard yesterday about aspirin, salicylic acid. That's a metabolite. And so poplar is rich in metabolites, and those metabolites affect a plant's ability to withstand drought tolerance. So if you build up metabolites, the leaves can withstand osmotic stress and are maintained on the plant. And so Tim, in his profiling of thousands and thousands and thousands of poplar samples, linked his phenotypic information with our GWAS information, and we've been able to link metabolites to drought tolerance to the genome. And so we now know genes that control that attribute. And Tim's here in the audience. Now enter the picture with Jay and Wellington, and we really hit the ground running here. We took the common garden GWAS population and identified genes that controlled carbon flux in the lignin pathway. There's a gene called EPSP synthase. It's the 6th enzymatic step in the shikimate pathway. 5,000 publications have been issued on this gene. It's the target of Roundup. So when you spray your plant, Roundup interferes with the function of EPSP synthase. And so nobody had ever noticed that there's a small DNA co-option, a helix-turn-helix piece of DNA that came from a bacteria that evolutionarily poplar obtained, and it entered into a regulatory function. So EPSP synthase regulates the flow of carbon away from the flavonoid pathway and into lignin. And so we could begin modifying lignin based on the expression of that gene. Mycorrhizal fungi have beneficial effects or beneficial impacts on plant growth. Poplar has a relationship with Leucaria. This fungus forms an intimate association with the roots of poplar in the lab, but also out in the field. And we wanted to understand the genes that control that. It turns out that there's a single gene called a lectin receptor kinase, which regulates downstream gene functioning, that in the presence of one molecular form, one allele, you could get colonization. If there was an alternate protein form, no colonization would occur. We identified. Steve, can I drink out of your glass? I've been drinking out of your glass for 40 years, so that explains a lot. We cloned that gene and put it in Arabidopsis. Now, Arabidopsis is this 30-day herbaceous plant that completes its life cycle in 30 days. There are no known mycorrhizal associates in Arabidopsis. This single gene, when cloned into Arabidopsis, you could see the fungal hyphae here in wild type. The fungus didn't even recognize Arabidopsis. It grew over the surface of the roots. But with this one gene overexpressed, it began to form a mantle. And then it developed a Hartig net. And this Hartig net is a combination of fungal hyphae in green, intercalating between poplar cells and exchanging nutrients. And so by introducing just a single gene from poplar into Arabidopsis, we changed the entire physiology and behavior, which tells us now when we inoculate field sites in forestry with mycorrhizal inoculum, that mycorrhizal inoculum disappears because the local fauna and flora outcompete this. But by tuning the host with the fungus, we can now ensure that these field inoculations can be successful. And this work was also contributed by Jesse and Jay. Jay's here somewhere in the room, has had a wonderful impact on this whole field. We continued to work, Jay and Wellington, and we wanted to look at the difference, what genes control rough bark versus smooth bark in poplar. If you have smooth bark, you can chip the tree and put it in an anaerobic digester, and it performs well. But if you whole tree harvest and chip rough bark, the bark is a natural antibacterial barrier in nature. And so that ends up killing your bacterial bioreactor. So we wanted to find the gene that favored this and disfavored that. And it turns out that there's a single gene, it's a protein, that causes a hairpin or like Velcro structure. So in rough bark trees, the Velcro held the dead cells next to the dead cells, and that leads to rough bark. But the smooth bark, the protein has a single amino acid substitution that causes the protein to flatten, and therefore neighboring cells slough off. That is wild type in humans. The reason we have smooth skin is that exact same protein, the exact same protein in poplar causes a disease in humans called keratoderma, where the protein has mutated and forms hairpin. So we collectively are not all that different at the DNA level or at the protein level or the physiology level. than a poplar tree. And I think that insight is incredibly important. Sometimes we think we're at the apex of evolution. You know, natural selection has led to us. But in fact, we are just one of many, many, many organisms that have successfully survived over time. Thank you. The LSD was starting to get to me. So, yeah. We just recently found a new protein called Booster. This is recently published. Booster's another gene in poplar that acquired a segment of DNA from a bacteria and a segment of DNA from an ant naturally. This horizontal gene transfer occurred evolutionarily a long time ago. So we talk about genetic engineering and the fear of introducing unusual or unintended consequences. Well, this process of acquiring pieces of DNA from other organisms is occurring as we speak in nature. And so we found this gene, and this gene also has a piece of the chloroplast rubisco, which is a part of the photosynthesis apparatus in all plants. And what this gene does is promote photosynthetic quenching. So when a plant comes out of darkness and the sun rises on the horizon and light impinges on the leaves, photosynthesis initiates. And the rate at which that happens is determined genetically. And the height at which that happens is determined genetically. So the faster it can reach its asymptote, the more productive it is. The more carbon that's fixed, the more carbon that's available for growth or defense. So this gene regulates photosynthetic quenching. If it's present, it happens rapidly. If not, it happens slowly. We cloned this gene out of poplar trichocarpa, and thanks to CJ's work, put it into 717, the genotype that's used in transformation around the world today. And you saw increases in 30 days associated with this gene. We cloned it out of poplar, put it in Arabidopsis, and you see the same effect in Arabidopsis. We've now put it in tobacco, soybean, rice, switchgrass, miscanthus, and it has the same effect in all those organisms. This is being licensed today by Living Carbon, a carbon sequestration company, and by the Gates Foundation to put it into cereal production in Africa. And so it is a single gene that has a profound effect on photosynthesis that we hope ultimately will have economic value out in the field. Let's see. So what you're looking at here is our new phenotyping laboratory, and I know you've built these in Umeå as well. The pinch point on information is no longer genetics or genomics, it's phenotyping. So how can we collect or accumulate massive amounts of information and relate that to performance? So this is the GWAS population, not in a common garden, but now in a greenhouse where we use hyperspectral imaging. We have 5 modalities, 2 hyperspectral cameras, a near IR camera, a chlorophyll fluorescence camera, and a thermal camera that happen inside these chambers. And so you can accumulate a terabyte of information in a week's period of time. And that information looks like this. And John, Mirko, and Dan are computational biologists. This is a single genotype on a single day with alternate spectra from the hyperspectral cameras. And now what you're seeing is that although we view a plant as homogeneous, its response to drought occurs everywhere, or its response to nitrogen limitations occur everywhere. That's not true. You see these subtle areas within the plant, and we can get a pixel by pixel assessment of that, where we now know that certain traits, certain behaviors occur at the tissue level, not on the whole plant level. And we can identify the genes that are associated with these spectra, and these spectra can be associated with things like nitrogen use efficiency, water use efficiency, bacterial infection, mycorrhizal association. And in a matter of months, we can run entire populations through the greenhouse and identify genes to trait associations. So I think we're going to see a lot more of this high-throughput phenotyping, not only in the greenhouse, but in unmanned aerial vehicles, flying over our forests and characterizing behaviors on broad swaths of land in nature. I talked about cell-level expression. So there's a new technique, single-cell genomics, where you can isolate the nuclei out of a single cell. Steve and I used to have to grind up whole plants, get this homogenized set of disrupted tissue, and then run it on a few markers. Now we can sequence individual cells from meristems, root caps, root hairs, the cortex, the xylem, the endodermis, and the second endodermis. And with that, we can identify not only networks of genes that are expressed at the tissue level or the cell level, but we can find promoters that are associated with that network of expression. And then we can take those promoters, and instead of doing constitutive overexpression of a gene on the whole plant, we can use that tissue level or cell level promoter to only turn the gene on in the tissue at the time we want it turned on. And this will revolutionize how we approach transgenics in the future and CRISPR-Cas9 modifications. With that, we've now sequenced 88 new genotypes, full-length, long-read genotypes, and we created what's called the pan-genome. This is variation at the DNA level amongst alternate individuals in a population. So I told you, or maybe I didn't mention, maybe last night I did, that it was a long time ago. I can't seem to remember. It's the LSD, I'm sure. Poplar has about 45,000 genes shared among all the individuals we've sequenced. But when we did long-read sequencing in Poplar, we found that there's this proportion of the DNA that's unique to individuals. So in fact, Poplar has about 75,000 genes. You have the 45,000 shared among everybody, and then these unique genes that are only found in certain individuals. So we can leverage that information now in what's called structural variants. Instead of SNP, we're looking at whole chromosome deletions or whole chromosome insertions and identify or associate genes to phenotypes that way. And we use computational AI-driven approaches to do this. And then finally, we need to get better understanding of expression. And what you're looking at here is a technique developed by Shohan, where you can take any gene that you're interested in. And you don't have to have a transgene. You can use this CRISPR-based system to look at where that gene is expressed. And so no stable transformation involved here. It's based on green fluorescent protein and guide RNAs that bring a GFP protein together. And so what you're seeing here are individual genes and where they're expressed at different ratios. And so we can use this technique to then also verify what we see here at the DNA level. This is the expression level, and this is the genome level. So with that, I want to thank my sponsors. None of this could have been possible without research support. And the Department of Energy in the US has been a wonderful supporter of my programs and the programs of many, many others. DOE founded the Joint Genome Research Institute, funds my current research institute, the Center for Bioenergy Innovation, and I'm in debt to them for their 20 years, 30 years of support. And with that, I want to thank you for your attention and just leave you with this one thought. If you, as early career scientists, surround yourself with brilliant, hardworking people, good things will happen. I've had the benefit and privilege of working with dozens and dozens of hardworking scientists who elevated my science greatly, and I wouldn't be here today without them. As early career scientists, do the same. Find those bright, hardworking people, collaborate with them, and you too one day might be standing here on this stage. So thank you very much. Thanks, man.
Absolutely fabulous. Thank you. I could tell that neither you nor Steve are microbiologists. immediately when you took that glass, and there was a comment about LSD, and I don't want to say any more than that.
It was a joke.
I thought only about microbes. I'm a microbiologist, as many of you know. We have a minute or two,
Okay.
and as you were talking, I was thinking about early days of doing PCR with a hot water bath and a cold water bath, which we did. Hard to believe for the young researchers
And
today.
physically moved it. Yes.
Absolutely. We had a name, a person's name for the machine.
Yes.
But a huge driving force, and it would have come out from the shotgun sequencing, has been machinery. So if you read, which I'm sure you have, Craig Fenter's book, Genome Decoded, it's fascinating. If young people should read this book, he talks so much about technology, the access to the machines. So what do you think about that? When you started, were you thinking, I mean, he was going, I think, he linked up with ABI and got every first machine they built, and they broke, and some crazy stuff.
The technology is just amazing. You know, the first PCR machine that allowed me not to move reagents from one water bath to another over a 24-hour period was the size of a small vehicle, and you had to put that in your lab. Now they're modular, and you can pick them up and move them, and my lab has 20 of them. And so not only the compactness, but the affordability. Same thing on sequencing. Those machines now are affordable, and postdocs in my lab can sequence an entire bacterial genome in a day.
And on the lab bench.
Right. So the technology has really evolved, and it will continue to evolve. I think AI and computer-based technology will play a big role in the next 15 years. So all of you that still have the opportunity to educate yourselves, become familiar with computational biology and computer power.
Yeah, it's great. Congratulations again.
Thank you.
Wonderful.
It's been a
celebration
pleasure.
of science.
Yeah.
Thank you for getting us started.
Yeah.
Thank you so much.